Hello, everyone. It's my pleasure to introduce our research. My topic is characterization of microRNA profiles in the mammary tissues of dairy cattle in response to heat stress. My name is Qiuling, and I come from College of Life Sciences, Langfang Normal University in China. The contents include 5 aspects: background, objective, materials and methods, results and discussions, and conclusions. Heat stress negatively impacts all features of dairy cattle production, including milk production and composition. It influences cow growth and development. Reduction.
Firstly, please, I will
Okay.
disrupt 1 minute.
Okay. Reduction in reproductive performance of a dairy cow is associated with
Firstly, please, I will disrupt for 1 minute for PowerPoint.
Okay. I can't share my slide.
Okay. Again, please.
Okay. Okay. Okay.
Sorry.
Okay, let's go on.
Okay.
Reduction in reproductive performance of lactating cows is associated with heat stress in summer. Heat stress causes cow metabolic disorders and decrease the immunity and increase the susceptibility to disease such as mastitis and endotitis, even to death in some severe cases. So we can say heat stress damage of Chinese Holstein cows means economic loss. Therefore, identifying and selecting animals that are thermotolerant may be the best way to alleviate the effects of heat stress. Non-coding RNA function as integral components of heat shock response in animals. The heat shock response is typically characterized by the rapid traumatization or activation of HSF1, that is heat shock transcription factor 1, and the subsequent transcriptional induction of heat shock. For example, HSPA1A and HSP90. We can see the figure. This model illustrated the diverse function roles of non-coding RNAs play or likely play in heat stress. To investigate the role of microRNA in the heat stress response, microRNA expression in bovine mammary glands were characterized by next-generation sequencing during summer and spring with and without a heat stress challenge. The experimental animals were 8 Chinese Holstein cows with the 4th lactating. The 4 Chinese Holstein cows were designated as biological replicates. They were divided into 2 groups, NHS group and HS group, from 2 different seasons with the different environmental seasons, with the temperature ranges from 15 to 20 centigrade, that is in March. We designated as non-heat stress group, that is NHS group. And the temperature ranges from 30 to 38 centigrade in July. We designated its heat stress group, that is HS group. After euthanasia, the mammary gland tissues were collected and frozen in liquid nitrogen until total RNA extracted for RNA's sake. Characterization of the stress response. Stress response of the mammals was characterized by a heat stress indicator and recording physical parameters. The temperature humidity index, that is THI, was as a stress indicator. Heat tolerance coefficient, that is HTC, and rectal temperature, that is RT, were physical parameters. The THI and HTC were recorded according to the formulas. From this table, we can see the physiological parameters recorded during different environmental temperature. We can see the THI of NHS group is 83.8, while the THI of NHS group is 65.8. Generally, the temperature humidity index, that is THI, is widely used in assessment of thermal burdens in cattle, of which that the value exceeding 72 indicates suffering of cows from heat stress. The rectal temperature and heat tolerance coefficient were also significantly different from the HS group and NHS group. We can see the rectal temperature of HS group was higher, and the HTC of the HS group was lower. So rectal temperature and HTC data also showed the HS group cows were in heat stress condition. And in each group, the RNA was extracted for RNA's sake. This table showed a summary of the statistics of microRNA-seq data. 8 small RNA libraries were constructed. A total of about 21 million high-quality reads were generated. Among them, about 20 million clean reads were obtained. after adapter trimming. Alignment with miR base indicated that microRNAs were highly enriched in all libraries. A pie graph showed relative abundance of different classes of small RNAs to assess the efficiency of high throughput sequencing for siRNA detection. All sequence reads were annotated and classified by alignment with R5 databases. Of the 18 to 30 nucleotide siRNA fractions, about 4 out of 5 were designated as other non-coding siRNAs, and only a small number aligned with small RNAs, tRNAs, siRNA, snRNAs, and rRNAs. We can see this is the frequency distribution of detected small RNAs, 18 to 30 nucleotides, based on all reads. From this figure, we can see the major reads from small RNAs were 20 to 24 nucleotides in length, accounting for 91.14% of total number. And the dominant reads of small RNAs were 22 nucleotides in length, followed by 21 and 23 nucleotides, and lastly, 20 and 24 nucleotides. Top 8 expressed microRNAs in bovine mammary gland was shown in this table. For example, BTA miR21-5P accounts for 9.15%, and BTA miR26A accounts for 5.46%. Together, they account for 35% of the total reads. To determine whether these small RNA sequences are genuine bovine microRNAs, we scanned bovine genome for hairpin structures comprising the candidate microRNA with miR-DIP2 software, which can be used to identify both known and novel microRNAs from deep sequence siRNA libraries. In total, we identified 483 known microRNAs and 139 novel microRNAs. The figures show the first nucleotides pairs of 18 to 30 nucleotides microRNA candidates. From the left figure, we can see the RNA that is U, represented by blue, was the most common at the 5 ends of the 20 to 24 nucleotides microRNAs in the mammary gland. And the U nucleotides also showed by blue. The U nucleotide was the dominant first nucleotide, while the G represented by red. The content was very low at position 1. The U nucleotide had a low frequency in the twice, third, and fourth positions. Differentially expressed microRNAs were analyzed based on sequence counts. In total, we observed 24 differentially expressed microRNAs between heat stress condition and non-heat stress condition. They were shown in the right table. We choose the 7 differentially expressed microRNAs based on RNA-seq to verify the microRNA expression. The expression of these microRNAs using RNA using qRT-PCR was consistent with sequencing results. To comprehensively describe the properties of genes and gene products, we executed gene oncology, that is goal annotation, and enrichment analysis from 3 ontologies, including biological process, cellular component, and molecular function. Target scan was showed that the target gene prediction showed about 27,000 genes that were enriched in 59 goal terms. These target genes may be regulated by the 24 differentially expression microRNAs. Goal function analysis showed that the predicted target of differentially expressed microRNAs were significantly enriched in different functional groups. For example, biological regulation, cellular process, metabolic process, regulation of biological process, response to stimulus, and a single organism process. KGG pathway annotation showed some signaling pathway may regulate heat stress in dairy cattle mammary gland. For example, TGF-beta, Wnt signal pathway, Notch, and Tau, MPK, JK-STAT. This indicated that the differentially expressed microRNAs identified in this study might act as a dominant regulator during heat stress. So in total, we identified 483 no bovine microRNAs and 139 novel microRNAs. Significant enrichment of predicted target genes of differentially expressed microRNAs were in several biological processes. And these data may be helpful for developing microRNA-based biomarkers for the control of heat stress in cows. This study was supported by some fundings, such as National Natural Science Funds, Science and Technology Project of Hebei Education Department, and Doctoral Foundation of Langfang Normal University in China. And these are our team members: Associate Professor Jia, Associate Professor Shang, Dr. Qi, and Dr. Zhang. That's all. Thank you for your attention.
Thank you.
Welcome to Langfang Normal University in China. I wish the conference a success. wish the conference a complete success. That's all. Thank you.
Thank you, Professor Lee. I hope in the future I will go to the Nanyang University.
Welcome.
Anyone have question? Please ask the question for Professor Lee. A question? Okay. Professor Lee, please show the slide about the miRNA, the last result. The result.
Pardon?
Okay. Okay, I will control. Okay. For this slide, I saw about the miRNA relate about the pathway. This pathway, I think this pathway relate to about the cell division. Yes.
Oh yeah, yes. Some signal pathway was significantly expressed higher or lower. So this signaling pathway may be play key roles in the mammary gland of dairy cattle. We want to study the pathway.
My research is about the cancer, but the cancer is related for this pathway and internet and surprising about the history relate to for this pathway again.
Okay.
Okay. I think it's maybe links between his stress and for your research and my research. Okay. Ajahn Panang, please.
Okay.
Okay.
Do you hear me? Do you hear me?
Okay.
Okay. In figure 14. In figure 14.
Figure 14.
In figure in this. Do you hear me? Do you hear me?
Yes.
Yes. In figure 14, that you mentioned that he showed in figure 14. Hit your slide in figure 14. Please. You show that in position about 20 to 24 is contain a lot of U. Okay. And the first position is contain U. I wonder what is the importance of this point. There are a lot of nucleotide U in position about 20 to 24, and the first position is U. What is the important about this? can you hear my question? In slide 14. Yeah, in this slide,
Okay.
it showed that in position 20 to 24, it contains U, and the first position is contain U. So what is important about this? Why this position is contain U? What is important about this slide?
Oh, the U was the dominant first nucleotide. And we can see the color. The U was the dominant nucleotide, and the right is A, green is C, G. So from 20 to 24, the first nucleotide bears is U, and the first glad at its position, for the first one, also U is the dominant nucleotide, and it is different at the 12th, 3rd, 4th, and 5th, etc.
Oh, okay. And I wonder what is important bias? Why you? Do you have any idea why you? So this is just like the bias, okay? It's contain you, a lot of you. But I wonder why you. Do you have any idea?
Oh, U is the bears of the first test. This is the common. Yes, it is the common in the first nucleotide.
Okay. Okay, thank you very much.
Thank you.
Thank you for Professor Lee. Keep a hand for Professor Lee again.
Okay.